Okay, in this video, this is the opposite of what we just saw in glycogenesis. This process is called glycogenolysis, and essentially this is just the simple breakdown of glycogen. So this is glycogen degradation. Now the question becomes, when would you want to release the stores of glucose that you have in glycogen? Well, we would want to do this under two conditions. One is under fasting conditions, and the other is under low energy conditions. So again, we have to look at the two primary organs that are responsible for storing glycogen: liver and skeletal muscle. Now, the reason we have to look at these individually is because the responses, the signals that release glucose from glycogen stores are different, and they're different for different reasons. So let's look at this in terms of the two organs separately. So here is liver, here is skeletal muscle. So again, as we started with glycogenesis, for glycogenolysis, let's start with skeletal muscle. Now, under fasting conditions, recall, indicate to us that we have high glucagon hormone levels in our blood. The reason for that is because our glucose levels in our blood have decreased from the fed state, and so insulin is no longer being secreted by the beta cells. What we now have are the alpha cells from the pancreas are secreting glucagon into the blood, which is an indication to the body that we are now in a fasting state. Now, very important to keep in mind, skeletal muscle's job is to produce ATP. There is no other reason that skeletal muscle has to respond to either a fed or fasting state except to supply itself with the substrates for ATP. So therefore, glucagon, being a signal that we are in a fasting state, is not going to have any effect on skeletal muscle. So skeletal muscle has no glucagon receptors. Skeletal muscle does not care that we are in a fasting state. Now, skeletal muscle does need glucose from its glycogen stores. And it's going to release this glycogen as glucose 1-phosphate. It does this by a phosphorylase reaction where we are going to be adding inorganic phosphate across the alpha-1,4 linkage of glycogen and releasing the monomer of glucose in the form of glucose 1-phosphate. Now the enzyme that does this is glycogen phosphorylase. This enzyme mediates the phosphorylase reaction of inorganic phosphate to release the glucose 1-phosphate. Once we have glucose 1-phosphate, we can convert this into glucose 6-phosphate by, recall, the phosphoglucomutase enzyme. So the same enzyme that converted glucose 6-phosphate to glucose 1-phosphate for glycogen synthesis can perform the same reverse reaction. Now, once we have glucose 6-phosphate, we can enter glycolysis and generate energy. So, from looking at this scheme, you can see that probably the most important regulation point is going to be at the initial release of glucose from the glycogen stores, and that would be regulating the enzyme glycogen phosphorylase. Now, there's three methods of regulation for glycogen phosphorylase. The first one is increasing AMP levels. So increasing concentrations of AMP are going to indicate that we have a reduction in energy charge in the cell. That means we're using our ATP. So how does this happen? Well, simply by muscle contraction. So with light muscle contraction and activity, we obviously burn our ATP for our myosin-actin interactions, and that's going to increase AMP. Now AMP is an activator, an allosteric activator of glycogen phosphorylase. So as our energy charge starts to decrease, we indicate to the enzyme that we need to release more glucose to generate ATP from glycolysis and the TCA cycle. The second level of regulation is increasing concentrations of the divalent cation calcium. Now, where does calcium come from? Recall the nerve impulse that generates muscle contraction is going to be releasing calcium from intracellular stores. This calcium is essential for myosin-actin interactions to initiate this process, and so the nerve impulse does this, but it also increases the calcium concentration that also acts as an allosteric activator of glycogen phosphorylase. So again, the muscle is undergoing contractions, using ATP, increasing AMP, and the calcium that is responsive to the nerve stimulus that initiates the contractile event. Both of those are positive inducers of glycogen phosphorylase to get the process going, knowing that ATP is going to be needed. Now, the third method is another endocrine method. And this is by the small hormone called epinephrine. Old term for this is adrenaline. What epinephrine is going to do is this is going to bind to its receptor, and much like the glucagon receptor, it is going to activate the heterotrimeric G protein. Recall, this is the activation of G-alpha, dissociation from the beta and gamma subunits. That will activate adenylate cyclase. This would be the G-alpha activated subunit, activates adenylate cyclase. This, of course, generates large amounts of cyclic AMP in the cytosol, and then this small molecule effector activates protein kinase A. Once protein kinase A is activated from this cascade of epinephrine, then this protein kinase A can further activate the glycogen phosphorylase. Now, why do we have three separate steps here for activation of glycogen phosphorylase in skeletal muscle? Well, simply because we are ramping up the activity of glucose released from the stores. So when we have small amounts of muscle contraction, we're generating a little bit of AMP. We want just a little bit of glucose released to regenerate our ATP. If we have a little bit more activity that requires more nerve impulses to release more calcium, this will be an additive signal on top of the AMP to turn up the glycogen phosphorylase a little bit more. Now, epinephrine is an adrenal response hormone. So this is going to be involved if we are into a fight or flight reaction. So under these conditions, we want to maximally turn on glycogen phosphorylase. So the signal of epinephrine going through the cyclic AMP pathway is going to be additive even more on the normal pathway. So now we get full activation of glycogen phosphorylase. Simply, if we are in a fight or flight response, we are going to be maximally using our ATP for myosin-actin interactions, and so we need to make sure that our ATP stores are not depleted and restored from our glycogen sources. So each one of these signals is slowly increasing the activation of glycogen phosphorylase. Now let's take a look at liver. The job of liver is to restore glucose levels in the blood. So the glucose that is stored as glycogen in liver is not going to be used by the liver cells. The glucose is going to be released by the liver from the glycogen stores and put into the blood to maintain glucose levels in a fasting state. This is just the opposite of skeletal muscle. Skeletal muscle is only concerned with obtaining the glucose from the stores for its own ATP production. The liver is not going to be using this, but the liver is satisfying other tissues and cells in the body for their glucose needs, primarily the brain. So the glycogen is going to be released in liver as glucose 1-phosphate. And of course, you can imagine this is going to be done by the glycogen phosphorylase enzyme, same enzyme that released the glucose in liver and muscle. And it's going to be using inorganic phosphate again. So this is a phosphorylysis reaction to release the glucose as glucose 1-phosphate. Now, same response as in muscle. Our mutase enzyme will convert this back into glucose 6-phosphate. So this is our phosphoglucomutase, same enzyme. But now we have a different path for the glucose 6-phosphate. In skeletal muscle, this went on to glycolysis. In liver, we are going to convert this into glucose. And the enzyme that does this is called glucose 6-phosphatase. Glucose 6-phosphatase. So this is going to remove the inorganic phosphate from the number 6 carbon of glucose, resulting in the production of free glucose. Now, recall that the GLUT2 transporter is always on the surface of the liver. This is not an insulin-responsive transporter, so it's always there. It works by high concentrations of glucose because it's a low-affinity transporter. So what's going to happen here is that as the glucose concentrations begin to rise, because we are releasing it rapidly from glycogen, it will now reverse itself and put itself into the blood and pass through the GLUT2 transporter. The primary organ that the liver is supplying, of course, is the brain. The major source of energy for the brain is the oxidation of glucose. The brain cannot oxidize fats very efficiently, and so glucose is the primary energy source. Now the question is what regulates this process? Well, simply, if the liver is responsible for maintaining glucose levels in the blood, that means glucose levels are going down in the blood, and that will occur under fasting conditions. So recall, under fasting conditions, we have the hormone glucagon secreted by the alpha cells in the pancreas, Now the glucagon is a signal to the cells where the body's in a fasting state because glucose levels are dropping. So it makes sense now that glucagon will stimulate the liver into action. And that action is releasing glucose into the blood because our glucose levels are going down because of fasting. So recall the signaling is very similar to epinephrine. Glucagon binds to its receptor. The receptor activates heterotrimeric G protein. Predominantly, this is G the alpha. G alpha subunit dissociates from the beta and gamma. The G alpha subunit then activates adenylate cyclase. Adenylate cyclase increases the concentration of cyclic AMP, and cyclic AMP then activates protein kinase A. And you already know what happens with protein kinase A because it's the same event that happens in skeletal muscle. Protein kinase A will phosphorylate glycogen phosphorylase, so it is becoming active. Glycogen phosphorylase activity then releases the glucose from the glycogen stores. Now, another side reaction to make a note of is that the protein kinase A is also going to phosphorylate glycogen synthase in both liver and skeletal muscle. Now, why is this important? Because if glycogen phosphorylase is active and you're generating glucose 1-phosphate from glycogen, what if glycogen synthase was still active? That would cause the glucose 1-phosphate to be reincorporated into glycogen and defeat the purpose of the activation of the counter enzyme. So the phosphorylation of glycogen synthase inactivates the enzyme and thus prevents what is called substrate cycling. Substrate cycling is if you have two enzymes that perform both reactions in opposite directions, two enzymes in opposite directions, such as the phosphorylase and synthase. If both of them were active at the same time, the glycogen would be converted to glucose 1-phosphate, and the enzyme going backwards would convert it back into glycogen, and you'd have a futile loop in there. So the protein kinase A is responsible for activating the glycogen phosphorylase under fasting conditions, responding to glucagon signaling in the liver, and it also inactivates glycogen synthase to prevent the futile loop. Same event occurs in skeletal muscle, except remember skeletal muscle is not responsive to glucagon. It is responsive to intracellular energy charge and the fight or flight hormone to make sure that the skeletal muscle cell is ready to flee any kind of dangers in response to the adrenal gland.